Hello and a very warm welcome to everyone joining us for today's Select Science webinar titled Compliance Ready: Intact Mass Analysis of Modified Oligonucleotides and Impurities using the BioAccord LCMS system. My name is Carrie Haslam and I'll be moderating today's session. I'm delighted to introduce today's webinar speaker, Dr. Katalin E. Doninu, Principal Chemist at Waters Corporation. So without further delay, I would like to hand over to Katalin for today's presentation, and I would like to thank him again for presenting with us.
Hello everyone, my name is Katalin Doninu, and today I'll be discussing a compliance-ready intact mass analysis workflow for analyzing modified oligonucleotides and impurities using the BioAccord LCMS system. Here is an overview of my presentation. I'll briefly discuss the main feature of the BioAccord instrument. Then I'm going to demonstrate the intact mass analysis workflow with molecular weight confirmation using a wide variety of modified and unmodified oligonucleotides. These compounds include the Waters MassPrep OST standard mixture, which contains 5 oligonucleotides, deoxythymidines. Then I'll show some examples with larger oligos, a 57 mer and a 100 mer oligonucleotide, and I will be discussing then several oligonucleotide modifications, most of them common type of oligonucleotide modifications encountered in the pharmaceutical industry. So here is a picture of the BioAccord LCMS system. Starting from the left, we see the Acuity UPLC iClass Plus, which contains the BSM binary solvent manager, the sample manager, the column compartment, and the TUV. Next to this stack, there is the RDA time of flight detector, which is a small footprint instrument. And next, we have compliance-ready informatics software that's used for instrument control and for data processing. There are 2 unique features of the BioAccord system: automated setup and intelligent diagnostics. All common tasks needed for instrument setup, including calibration, tuning, resolution check, are set up automatically. In front of the instrument, there are 3 solutions containing the calibration solution, the wash solution, and the log mass solution. In addition, instrument contains a built-in health system that monitors for problems and provides guidance on resolving them. As you can see from the front panel, there is a green light indicating that the instrument is ready or running samples. Should there be any issue with the instrument, the front panel will display a yellow check light. In the same time, there will be an icon displayed in the Waters Connect platform. In this case, the system detected a solvent leak. The user is then expected to follow some simple instructions to resolve the situation by clicking on the Resolve button. There are several BioAccord supported applications, including intact mass analysis of oligonucleotides that I will be discussed today. In addition, intact mass analysis of proteins, including antibodies or subunits, peptide mapping, release glycans, and peptide multi-attribute monitoring are also other applications supported on the BioAccord LCMS platform. The experimental conditions used throughout this webinar are listed here. The mobile phase contains 7 millimolar triethylamine for solvent A, 80 millimolar HFIP hexafluoropropanol in DI water. Solvent B contains half of these concentrations, 3.5 millimolar triethanolamine and 40 millimolar HFIP. All separations were done on the Waters OST column, 2.1 by 50, and the column was kept at 60 degrees and operated at a flow rate of 0.3 mL per minute. I would like to mention that we recently introduced a new column, a premier column for the OST column, and I'm listing the part number here. The premier columns contain an inert surface that prevents metal interactions of analytes and the column. The TUV detector was used in this application with a wavelength of 260 nanometers. The MS experimental conditions are listed here. The BioAccord system can acquire data in full scan or can acquire data using an MSE scan. In this case, there are 2 consecutive scan events. In the first one, only the precursors are detected. In the second event, In the second scan, the fragments of those precursors are recorded. In this case, for these applications for intact oligo analysis, we recommend using FullScan application, and the data was collected using FullScan. There are different mass ranges in positive and negative ionization mode, as listed here. The mass range that was used in this case is from M/Z 400 to 5000. The instrument has different acquisition rates in the range of 1 to 20 spectra per second. The mass resolution is constant, better than 10,000. And as you will see from the data presented here, typical mass accuracies are in the range of 1 to 15 parts per million. Here are some chromatograms recorded on the BioAccord LC-MS platform. So we have first an example with the OST oligonucleotide standard mixture containing 5 major components: deoxythymidines, 15, DT15, DT20, DT25, DT30, and DT35, as you can see in these 2 chromatograms. The top, it's a UV chromatogram recorded at 260 nanometer, corresponding with a load of 10 picomoles of each oligonucleotide on column. The UV trace can be compared against the TIC trace. The low-level impurities are failed sequences from DT10 all the way to DT25. These are low abundant impurities, and as you can see, they are detected in both chromatograms, the top UV trace and the bottom TIC chromatogram. The spectra recorded for the top 5 major oligonucleotides present in this mixture are shown here. All these spectra display a bimodal distribution, showing a maximum for low charge states, -3 and -4, but to the left we have a maxima again for higher charge states, including -7, -9, or all the way up to -15. This is typical for ion pair reverse phase chromatography analysis of oligonucleotides. These spectra provide a wide range of charge states over a wide M/Z range. So in this way, we can achieve very good deconvolution to provide accurate intact mass analysis for all these components. Here we have an example of the -12 charge states of the largest oligo in this mixture, DT35 or 35 mer. As you can see, with 10,000 resolution, MS resolution, the BioAccord system is able to resolve the isotopic distribution of this peptide, and the monoisotopic peak can be detected here. This helps us to improve the mass accuracy for the deconvolution process. When we load lower amounts of samples, as shown here with 100 femtomoles on column, we can still maintain very good sensitivity between the UV trace and the MS trace. All the 5 major components are still detected even with 100 times less sample loaded on column, and this is visible in both chromatograms. This also means that the MS background in the TIC chromatogram is very low, such that even with small samples, with small amounts of compounds loaded on column, we can still clearly see well-defined sharp peaks. The previously acquired data was processed with Waters Connect. Here I have a screenshot of the report from Waters Connect showing all the components identified in this sample. For each component, there is a response, which means UV peak area. There is an observed mass, which is obtained as the deconvolution from the ESI spectrum, which is compared with the predicted mass or expected mass that's based on the elemental composition, and then a mass error is calculated automatically. In addition, for the example shown before, we have here listed all the abundances calculated based on the peak areas in addition to the average masses and the mass error. This table demonstrates that for a wide range of compounds, 26 oligonucleotides, the average mass accuracy was 5.3 ppm based on all these individual measurements. Moving on from unmodified oligonucleotides, here I have an example of a 25 mer fully phosphorothioate nucleotide. In this case, 26 oxygen atoms from the phosphate backbone were replaced by sulfur atoms, and then we have a fully phosphorylated oligonucleotide. As you can see, we got a very good chromatography, good separation, good signal intensity in both UV and the TIC chromatogram. The spectrum, ESIMS spectrum of the 25-mer oligonucleotide is shown here. Again, we see the same features, the bimodal distribution, typical for ion pairing reverse phase chromatography. And when the spectrum was deconvoluted using base pair deconvolution in Waters Connect, error was 1.5 ppm. Larger oligonucleotides can be analyzed as well on the same biocord using pretty much the same experimental conditions, same column, same mobile phase, with slight method development to adjust for the gradient conditions. Here I have an example of a 57-mer oligonucleotide. We see the UV and the TIC chromatograms. Again, we got very good sharp peaks. The ESIMS spectrum of this compound shows a wide range of charge states from -9 all the way to -22. And upon deconvolution, the result indicated in terms of mass accuracy is shown here, 19.4 parts per million. Even larger oligonucleotides can be analyzed. These are about the size of the guide RNA used in CRISPR applications. So here is an 100-mer oligonucleotide, unmodified, and what we have is the UV chromatogram and the TIC chromatogram as shown in the previous examples. And the spectrum of this larger oligo is shown here. Again, it's a variety of charge states from -19 all the way to -36. And upon deconvolution, the result indicated in terms of mass accuracy is close to 27 ppm. In the next section of my presentation, I would like to show some examples with extensively modified oligonucleotides. If we look at the basic structures of the oligonucleotides, the 2' position usually is the one that's typically modified. So in this case, the R1 functional group was modified either by a fluorine atom, all-methyl group, or a methoxyethyl functional group. In addition, we have some examples with phosphorothioate. As I mentioned before, when sulfur atom replaces an oxygen in the phosphate backbone. Finally, we have some modifications on the 5' end, including a GalNAc sugar or a flavridine amidate replacing the OH functional group. There are several modifications. The ones are listed here, plus the locked nucleic acids, as you'll see in the later example. In all these examples, we use and compared an unmodified oligo against the modified oligo. So in the same chromatographic run, we recorded the spectra for both of these compounds, and then we compared the mass accuracy. So the first mixture analyzed was an oligo-1 mixture containing 3 oligonucleotides. The first one, labeled as oligo-1 control, is a 20-mer that has no modifications. We can calculate the average accurate mass based on the elemental composition for this oligonucleotide. A second oligonucleotide has exactly the same sequence, but it contains a tag, a GalNAc sugar attached via C3 linker to the 5' end of the molecule. Finally, we have an oligo-1 test compound containing 21 nucleotides, and only 8 are left unmodified. 13 others are modified with different functional groups. For example, if we look at the structures of these modifications, we have 6 cytidines that are modified in the 2' position by methoxyethyl. Then we have 1 adenosine that's modified with the same functional group to methoxyethyl. In addition, 2 guanosines highlighted in red in the sequence are modified with the same functional group. There is only 1 methyluridine, and there are 3 methyl cytidines in this structure highlighted in brown. As you can see from all these modifications, the molecular weight of this oligo is by over 1,000 Daltons. The chromatographic separation of this 3-component mixture is shown here. We have the UV trace and the TIC chromatograms. The unmodified oligo, labeled as oligo-1 control, elutes first. Then the GalNAc modified oligo elutes second, and finally the heavily modified oligo-1 test compound is the last eluter in the separation. As you can see, by adjusting the experimental conditions, one can achieve very good separation of all these related structures. The spectra recorded for the 3 oligonucleotides are shown here, and again we see the same pattern with the bimodal distribution for all 3 of these compounds. In addition to that, what I displayed here in the inset is the result from Waters-Connect deconvolution of the 3 components. As we can see, we can obtain very good mass accuracies, 1.6 part per million for the unmodified one, minus 3.9 for the GalNAc, and minus 15.2 for the heavily modified oligonucleotide. A full screenshot from Waters-Connect is shown here, indicating the results that we've seen before regarding the mass accuracy, but in the same time, the UV and the TIC chromatograms are shown, the deconvoluted spectra are displayed as well. A second oligonucleotide mixture was analyzed. In this case, the unmodified oligonucleotide is a 28-mer, and the modified version contains a modification at pretty much every nucleotide. There are different types of modifications, including starting with the 5' end, which contains fluorescent amidate. Then there are 6 fluorocytidines highlighted in red in the structure, in the sequence. There are 8 O-methoxyadenosines, which are purple, 6 O-methoxyguanidines, 7 2'-fluorouridines, and then there is an inverted deoxythymidine at the end. When we look at all the modifications that we have for the 28-mer, we achieve actually a molecular weight increase of around 1400. This 2-component mixture can be easily resolved on the OST-C18 column, and the chromatograms are shown here. The first eluter is the unmodified oligo 2 control, followed by the oligo 2 test, the heavily modified oligonucleotide. In this case, the 2 electrospray spectra are presented in this slide. Again, we see the bimodal distribution, charge states anywhere between -3 to -12, and one can see that we can obtain good mass accuracy for the control unmodified oligo, 6.5 parts per million, versus the heavily modified oligo, -11.8 parts per million. Finally, we have here a mixture of 4 oligonucleotides. These are all 16 mers, and they contain a locked nucleic acid configuration. So again, we start with an unmodified oligo labeled as oligo 3 control. Then we have 3 locked nucleic acid structures close to the 5' end, and other 3 locked nucleic acid structures close to the 3' end. The 3 T's, these are locked nucleic acid thymidines. In this case, the same 2' position is linked through the ring, to the sugar ring via a methylene group. So we have locked nucleic acid thymidines and 3 locked nucleic acid guanosines. In addition to this locked nucleic acid modification, we also have phosphorothioates, and it's a fully phosphorothioated oligo, shown here as oligo 3 PPT phosphorothioate control. And then finally, we have a structure that contains the phosphorothioate and the locked nucleic acid configurations. So all these 4 compounds were mixed and separated, and the results are going to be shown on the next slide. The TUV and the TIC chromatograms for the mixture are displayed here. What we can see is that we get very nice sharp peaks for the control compounds that do not contain the phosphorothioate, and then we have broad peaks, later eluting peaks as oligo 3 PPT control and oligo 3 PPT test. And these are broader peaks probably because they contain diastereoisomers due to the many sulfur chiral centers due to sulfur atoms. I'm going to compare in pairs the spectra recorded for these compounds. So here we have the control 16-mer oligonucleotide against the phosphorothioated compound. The spectra look very similar, and the difference between them is that in the case of phosphorothioates, we have to use special isotopic model called phosphorothioate oligo, because sulfur has a different isotopic abundance, has a unique isotopic abundance. So therefore, we have to use a different isotopic model. In the case of the regular oligos, we can obtain a very good mass accuracy upon deconvolution, 0.2 parts per million. And if we use the phosphorothioate oligo model for deconvolution, then we can obtain very good mass accuracy for the modified oligo. If we don't use the PPT model and we use a regular oligo model for a phosphorothioate, then we don't obtain very good mass accuracy as shown in gray. The mass accuracy is minus 38 ppm. So definitely we really need this PPT isotopic model in order to obtain accurate deconvolution data. The second pair of oligonucleotides is shown here. Both are modified with locked nucleic acid configurations, but the spectrum on the bottom corresponds to a phosphorothioate. Again, if we use the proper model, we obtain very good mass accuracy for the heavily modified phosphorothioate, 7.2 ppm, and we also obtain very good mass error for the regular oligo, minus 4 ppm. To summarize, I included a table here where we have all 8 compounds, modified and unmodified, according to their, highlighted here, according to their retention times, their structure, the average mass, and the mass accuracy. As we can see from the right side of this table, mass accuracies better than 15 parts per million were obtained for all these compounds, regardless whether they are modified or not. To summarize, we have shown some data for 7 different oligonucleotide modifications that were analyzed using the same OST column and the same mobile phase, just gradient optimization for separation of different mixtures. A typical bimodal distribution was observed for all the ASIMS spectra, regardless whether the oligos were modified or not. Typical mass accuracies were in the range of 5 to 15 ppm for both the control or unmodified oligos compared to the extensively modified oligos. So we demonstrated that we have here an automated compliance-ready LC-MS workflow that can provide a robust, easy-to-use solution for intact mass confirmation and impurity analysis of oligonucleotides. I have an acknowledgement slide here, and I'm more than happy to answer any questions you may have. Thank you very much.
Thank you very much, Katalin, for today's informative presentation. So it's now time for the question and answer session. So the first question we've got is, why did you choose this LC column for the analysis? Only low metallic impurities or others? Thanks in advance.
So we choose this column because this is a recommended column for oligonucleotide analysis. It works really well with ion pairing applications. There are actually 2 columns that I listed in my presentation, and one of them is a premier column, so it's a new column that was introduced this year, and this column is it contains inert surface that prevents interactions between analytes that could interact with metals. So we recommend using this column, although most of the work was done on the regular OST column.
Perfect, thank you. So on to the next question. So what we've got here is, would this oligonucleotide workflow work for conjugated oligonucleotides used in molecular diagnostic applications?
Yes, that's a very good question. So actually, there was one single example showing a conjugated oligo. I think it was part of the oligo 2 mixture. So it contained a fluorescent amidate tag that was attached to the 5' end of the oligonucleotide. So essentially, I've been running other oligonucleotides, conjugated oligonucleotides, including Alexaflor or TET labels, and there was no problem. So essentially, we can run different type of oligonucleotides, conjugated oligonucleotides as well using this application. We can obtain very good spectra and we can get very good mass accuracy.
Perfect, thank you. So on to the next question. So we've got, what is the largest oligonucleotide in terms of size and molecular weight that you have analyzed on the BioAccord platform?
So the examples that we have here, we ran an 100 mer and we got a really beautiful spectrum, and we can get mass accuracy of around 25 ppm. I've run larger ones, including up to 200 mers. Probably the largest will be 500 mer, 500 nucleotide. That's at least what I know in terms of the largest that was analyzed and was able to produce a spectrum.
Perfect, thank you. So another question we've got here is, are there other optical detectors available for this application?
This application actually was done with the optical TUV detector that goes all the way to 700 nanometers. But we have two other detectors, a PDA detector that's able to record the full spectrum, UV spectrum, goes all the way to 800 nanometers, and a fluorescent detector. So potentially we could use these other optical detectors if the compounds warrant such application. And if you want to either extend the visible range or use it to detect fluorescent molecules, maybe for conjugated oligonucleotides using diagnostic applications.
Perfect, thank you, Katalin. So the next question here is, were the experimental conditions modified significantly for each class of modified oligonucleotide?
So as I briefly described, the column was the same, the mobile phase was the same, but the gradient conditions had to be adjusted for each particular class of compound. So it does take a little bit of method development, especially since they are closely related structures. We have unmodified versus modified present in the same mixture. Sometimes if the oligo contains a lot of modifications, then it's relatively easier to perform the separation. Sometimes we have an oligo that contains only a GalNAc sugar tag at the 5' end. Even in such cases, when we have a single modification, we are still able to resolve the compound using ion pairing chromatography on this column. So the only thing that was really changed was the gradient profile. And that encourages us to say that probably we can analyze a wide variety of oligonucleotides, really regardless of their structures, using this same system with the same column and the same mobile phase, with just a little bit of adjustment for the gradient.
Great, thank you. So another question is, could you please explain the kind of analyzer that you could use for this range of oligonucleotides?
In terms of MS analyzers, we have a time-of-flight TOF instrument. The BioAccord is a TOF instrument, so it records fast MS spectra full scan. In this case, we recorded full scan MS spectra in negative ion mode over a mass range of 400 to 5000 m/Z. And this is perfect for measuring even higher molecular weight oligonucleotides, probably 100 mers, 200 mers, and so on.
Perfect, thank you. So on to the next question. So what we've got here is, what was the mass accuracy obtained with these 200 mer or 500 mer analyzed with the BioAccord?
So the highest example, the highest molecular weight that we run here was 100 mer. I did run 200 mers as well. The mass accuracy that you would expect would be around 30 parts per million, 30 ppm, 30.
Perfect, thank you. So on to the next question. So we've got, what mode will you choose to deconvolute a MAB siRNA?
Okay, this is a tricky question. So we use, there are two deconvolution models, two modes to deconvolute, MaxCNT1 and base spray. In most of these applications, actually in all these applications that I've shown today, I use base spray. And for larger oligonucleotides, sometimes MaxCNT1 provides better deconvolution. It really depends on how big this molecule is. I assume that MaxCNT1 could be useful in these cases.
Great, thank you. So we've got another question, which is, what precautions did you take for preventing DNase degradation of the sample or sodium adducts?
So sample degradation wasn't really a big problem because these are rather stable DNA type of oligonucleotides. However, we have some examples. I ran some mRNA molecules or RNA oligonucleotides, and they are more labile, and sometimes adding protease inhibitors or making sure the samples are analyzed right away, or I mean, there are always some precautions that you have to take. Regarding the adducts, we've seen that the quality of the reagents makes a big difference. So I think I was actually screening and looking at different HFIP reagents, and that looks like the major source of sodium adducts from my experiments. But the DI water is also important. So the quality of the reagents matters. The cleanness of the glassware matters as well. So there are ways to mitigate and try to reduce sodium adducts by making sure the mobile phases and the system is clean and doesn't have a significant amount of contamination.
Perfect, thank you. So we've got here, are there ways to modify the mobile phase, perhaps by post-column addition, to reduce the charge state distribution without adversely affecting the chromatography?
So that's a very interesting question, because what we see mostly with all these examples is this bimodal distribution. So we have some low M/Z charge states maxima around maybe -4, -5, and then we have another distribution peaking around -9 and -10. So if you want to deconvolute and get a precise measurement, actually you get a lot of charge states here over a wide range, and this helps. Actually, the base spray deconvolution process also helps because you have so much signal spread over the entire mass range. You have actually multiple measurements. So in some cases, this is beneficial. Really depending on the application. Sometimes you want to do reduce the charge states and get less signal, and definitely that's something that someone can do. I haven't tried that on these type of compounds, but we have done some other experiments, and we show that that's definitely possible, and you can reduce the charge states. And sometimes if you have maybe 3 or 4 charge states, maybe you can speed up the deconvolution process. I'm not sure exactly what the clear advantage would be. Maybe if you want to do fragmentation and pick certain charge states that fragment better, that could be an option. But yeah, this is an interesting approach.
Perfect, thank you. Another question is, which mode is best for intact mass analysis, whether to use sensitivity or resolution mode?
So this is more of a question for QTOF instruments like Xevo G2XS or SYNAPT, which have multiple resolution modes. On the BioAccord, we have positive, negative, and then we have either full scan or MSE scan. So we have 4 different acquisition modes, and there is no resolution and high resolution mode or sensitivity mode. The instrument resolution is fixed at 10,000 resolution. So in this case, the data was acquired full scan, negative mode, M/Z 400 to 5,000.
That's great. Thank you, Catalin. Well, that's all we've got time for today. So thank you to our speaker, Catalin, for today's informative webinar. And thank you to everyone joining us online and for sending in your questions. If we didn't have time to answer your question, we will follow up with these after today's event. If you do have any other questions, please feel free to submit them now using the Submit a Question tab to the left of your screen, or email me at editor@selectscience.net and I will follow up with your questions. You can also download related documents in the Related Resources tab to the left of your screen, including a certificate of attendance. If you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. Goodbye, and thank you once again for joining us.